Today I want to talk about what Covaris has done this past year to help standardize our end of DNA sequencing as we move more into a clinical environment. Okay. Covaris has been around for a number of years. We have a variety of instrumentation that is used for shearing DNA into very specific sizes. They all use ultrasound for the shearing technology. We have two M-series instruments and then our L-series instruments that may be very familiar to all of you who have been doing next-gen. We need our workflows to go smoothly and to involve very few places where human error can come into play. So we've made some changes in the instrumentation to help move through backlogs and to create a more uniform process. So just a little bit of background on the ultrasound. The ultrasonics is used to create cavitation bubbles within a sample, and those bubbles are used for applying mechanical energy, which will make anything smaller. So for most of us, that's DNA. Also RNA, if you need to fragment that for analysis and make it smaller. A chromatin is used in this very same process for chip work and chip-seq. It's also used to shake things loose, loosen things up, free them up, things that have been bound tightly either to maybe a protein complex or to a matrix, such as a dried blood spot, or to increase the yield of something such as cell-free DNA, which is historically, you know, we all know this, it's wrapped around histones and other protein complexes and a little bit hard to free up. We also use the same ultrasound energy and the same instrumentation for opening things up, such as cells, to release the biomarkers that we need. The ultrasound's non-contact, so there's no probes. It's all done in a closed tube environment. It's temperature controlled throughout the process, and it's highly reproducible. So just a little bit more background on it. DNA processing on the Covaris instruments is independent of the concentration of the DNA itself. It's also highly reproducible, so shearing runs overlaid, as you see in the middle diagram, are all on top of each other. And it's also highly adjustable as far as the sample sizes that we can produce with shearing methods. Anything from the very small end, 125 base pairs, up to 1,500 base pairs. If you need to go higher, we have other technologies that you can use to produce up to 5,000 base pairs in a uniform shearing. So in the last couple of years, we've been hearing from you, our clients, that we need to have our workflows improved to really suit the clinical environment. Also to suit an environment that is more highly automated, using more robotics. So we've done that this past year, and I'm here to tell you a little bit about the new system that we have out. It's called the LE220 Plus. And OneTube, which is a new consumable that works with the system. They both work together. These are designed for higher throughput laboratory situations where automation is desired. It uses an automated water level adjustment system, which is important for processing reproducibly. It's also important for eliminating user-to-user variation with the instrumentation. We also have a scheduling system now where instrumentation can turn on, say at 4 in the morning, you start degassing your water tank. 5 in the morning, you may start chilling your water tank, and when your team comes in, it may be 6 in the morning, everything is ready to go. A little bit more about the new system. Again, it's designed with the high throughput laboratory automation in mind. And the water level adjustment is one of the most important aspects of the system. Now, the system does include the new OneTube, which is a completely different type of shearing vessel than we've used in the past. Our past shearing vessels have all been glass-based, and they have all used an AFA fiber in the tubes to help produce the cavitation bubbles. The new tube is completely different. It's plastic. It's made out of a composite. And the shape of it has been changed too. It's now compatible with your common laboratory instrumentation, such as PCR machines, automated pipetting systems, your heat blocks, your mag bead stations. The DNA sharing still uses the same ultrasound, but the new vessels do not include the AFA fibers that have been used in the past as the source of nucleation sites for the cavitation. What we've done instead is engineered the inside of the tubes to be a source of cavitation bubbles themselves. Now, as DNA sequencing has matured, we've gone to lower and lower sample volumes to help accommodate the needs of the laboratories and also to match what is done with the popular library preparation kits. So the new consumables, one tube can shear samples down starting from 10 microliters in size and up to 50 microliters in size. The total volume of the tubes themselves goes up to 200 microliters, and this is important to downstream applications. In the past, we've sheared in our glass tubes and then removed the DNA for the next step in the processing because glass is not compatible with most laboratory processes. Okay. A little bit more about the one tube. The polymer is designed with low impedance. What this means is when the ultrasound moves through our water trough and into the tube, the energy moves through our one tube and into your sample. It's not absorbed by the container itself. Other plastics, this has been a problem. Heating would ensue from the energy being absorbed by the plastic. We've worked a way around that by developing the right kind of plastic. Again, the configuration of the tube is such that we are still producing the needed cavitation bubbles by having features inside the tube that create what we'll call a synchronized cavitation field. So when the ultrasound energy hits your tube, goes into your sample, bubbles form along the tube itself, grow as we send in the ultrasound, and then collapse as we go to zero energy states. This all happens very fast and is very, very reproducible. So what we've done is everything that you would have to do too, of course, is we wanted to show that the new process is highly reproducible. So we ran 48 samples and overlaid them with two different shearing sizes. And what I'm showing here is a 10 microliter volume with 175 base pairs in the blue. And the red shows 350 base pairs. Both of these are popular sizes for library preps. Okay. The new tubes also contain a little bit different sealing format. In the past, the tubes have all come with a foil seal on them, and you pipette through the foil seal and cover with a foil sheet. The newer tubes, the one tubes, have your choice of covering with a foil sheet, same as you've done in the past. But we also have strips and caps. Also, if you prefer to use sealing caps, you can certainly do so. A little bit more about the one tube. Again, doing some bridging experiments to compare the one tube with our microtube 50s, which have been used for most of the library preps currently on the market. We're showing is all steps can go on in the one tube, not just for the shearing, but also for the library preparation. The new plate, again, can fit on your thermocyclers and your pipetting workstations, and we're pretty much agnostic. So we do work with a lot of different automation partners such as PerkinElmer, Hamilton, TCAN, others that need support with the newer platform. Again, the new volume for the one tube is up to 200 microliters. This is a lot of room for doing the rest of the library prep, which here I'll show you a little bit of work that we've done with Swift using their S2 kit. We've done the shearing in our tubes and then the entire library prep system without removing the sample from the tube. So this saves you on sample loss and transfers and mistakes that would have been made during transfers. Okay, just to show a little bit more of the talent set of the one tube. This is working with an artificial microbial community where we've pooled 3 different types of bacterial genomic DNA, sheared in the AFA 10, the microtube 10 plates, with both 100 nanograms and 1 nanogram, and the results were identical. We also did some next-gen sequencing using the ApoE gene, and again, results were identical. And if we dig in this a little bit further, we'll see that the one tube actually performed a little bit better than our standard microtube plate. So the way we do this with the LE Plus is we've added an internal sensor that senses the top of the water in the 5-liter water bath and adjusts the height of the tube itself and adjusts the height of the transducer so that this is no longer something that is done by individual users. It's eliminated the human factor. We've also introduced scheduling and easier to use software, again, eliminating mistakes. Okay? Just to kind of wrap things up, this machine is also open to more automation. We do have the ability to add doors and a light curtain to the front of the machine. So if you need arms to move plates in and out, to add higher throughput to the instrument, you can certainly do that. Covaris, we're easy to get hold of. We are worldwide. Headquarters is in Woburn, Massachusetts, and we do have a large presence in Europe.